It's a great pleasure to introduce Jean-Michel Claverie. Jean-Michel is from CNRS in Marseille and has made a significant contribution, I think, to making Marseille a centre for infectious disease research and a centre for genomics. And he's going to talk to us this evening about his work on giant viruses. So, Jean-Michel, thank you.
So thank you very much for your nice invitation. And I must say that I thank you even more for your broad-mindedness to select me as a keynote lecturer in the context of an infectious disease meeting when all the viruses I'm going to talk about are on infecting amoebas. And so you can sit back and relax. Nobody is going to be harmed during the next hour for this talk about giant viruses. So this is a saga that actually started not long ago, about 10 years ago, and that accelerated tremendously the last 3 years. So a little bit of a history of science first. You all remember the germ theory by Pasteur, and that was very much also posted by Koch, that was actually linking disease to microbes. And for the first time, disease at some kind of a real cause. That was the end of the miasma theory, which was basically if something stinks, it's going to get you sick. But in fact, microbes were known before, about almost 2 centuries before. I always like to remember that Anthony van Leeuwenhoek is the real discoverer, is the real founder of microbiology. But because of the link of disease and microbes, immediately translational research picked up. And in a way to try to it was like the first biotech invention, basically. Immediately, Chamberlain invented a contraption to try to sterilize water. That was the well-known Chamberlain filter to actually try to remove bacteria from the water to avoid, for example, typhoid fever in Paris. And so this contraption, this protocol, became the golden standard for quite a long time to define if something was caused by a microbe. You had some kind of something happening, rotting, a disease, something happening, and then if that thing will survive filtering, it was not an infectious disease, it was not caused by a microbe. If it was stopped after you filtrate whatever the juice on the filter, it was caused by a microbe. That was so the best way to isolate microbes for a very long time. Until not long after that, because in 1892, that was exactly the date of the Jubilee, the 70th anniversary of Pasteur, when he was hailed as the biggest, the most famous scientist in France and in the world at that time. A totally unknown guy in Russia did an experiment. It was Dmitry Ivanovsky, when somebody asked him to find the cause of the disease called tobacco mosaic disease of tobacco. And what he did, he took some disease leaf, he made a juice out of it, and he did the golden protocol of his time, filtered the juice on the Chamberlain filter, and was expecting, of course, that nothing would happen. But then something happened. The new leaf got infected. Okay, so he discovered a new type of infectious agent. And pretty soon after that, it was discovered that many other infectious agents were in fact filterable. And they were called viruses a little after that by Beijering. Okay. And so that was the first discovery of a new type of agent that was called virus. But at the time, even microbes sometimes were called virus. But so that was filterable virus for a long time. But they remain mysterious because you could not see them and you could not cultivate them. So, of course, it takes a very long time. It takes basically the invention of the electron microscope to be able to see the first virus that was, again, TMV. So for about 50 years, nobody really knew what they were until, in fact, André Lvov was the first to put up real criteria, rational criteria to define, to discriminate between viruses and microbes or cells. And basically those criteria are all negative properties, which is not a good way of defining something when everything is doesn't have something. It's not a very, you already realize there's something wrong about it. It's not visible by light microscopy, so it's the same thing that it goes through the filter. It's much less than a micron. It's not retained by the Chamberlain filter. It goes through. It's not cultivable outside of cells. It doesn't have its own energy production, doesn't make ATP. They call that the Lippmann system at the time. And it doesn't have ribosomes that don't translate protein, which is a very important criteria even as of today. And it doesn't replicate by division. It doesn't divide. It just replicates, but no way, not by division like cells do. So those are the main criteria that we are still using today. You'll see to define that we have a virus in hand or not. And so the story of the giant viruses started about more than 10 years ago now, when during an epidemic of pneumonia in Bradford near Leeds, as usual, the person in charge, that was Tim Robotham, start going around in cooling towers and in water system to find what could be the cause of that Legionella disease, looking for amoebas, because normally, as you know, Legionella lives in amoeba, so you look into the amoebas to see if you see the kind of the causative agent of the epidemics, okay? And so he saw some kind of strange things. He saw those little dots that were, in fact, in a way taking the Gram stain, those little dots here, but that was definitely not Legionella, because as you know much better than me, that Legionella don't have that kind of shape. They are bacilles, they are like a rod shape. And so he was very happy because he thought he had discovered a new type of pneumonia agent, and he called that Bradford coccus because it was a round shape. Okay. And it stayed that way, so he started to try to characterize it, try to cultivate it in the regular ways, tried even to do some kind of a 16S RNA sequencing. At the time, he was one of the pioneer labs trying to do that, and they failed. But he was very smart. He did one thing that you all know you should never do. He didn't throw the sample away. He kept it in a fridge for a very long time. And that sample actually came out in the Didier Raoult's lab in Marseille about 10 years later, in 2002. And we are pretty close at that time to throw the sample away because, as you know, Didier Raoult is the world specialist of rickettsia. And so he tried all the tricks and trade to try to cultivate that intracellular parasitic bacteria. We failed, but just before we threw the sample away, somebody got the bright idea of looking at the sample under the electron microscope, and we found that this thing had some kind of very regular shape for a bacteria, which, as you know, is not always a good criteria because there are square bacteria, for example. Okay, so but we thought, okay, maybe this is not exactly what you thought it is. And in fact, this was the first giant virus. That was Mimivirus. This is a nice illustration by the new scientist. So this is the HIV, which we've just talked about, which is already quite a large virus for the size of genome it has, okay? And this is Mimivirus, and this is the classical, rhinovirus that we all are getting every winter. And so this was the first virus you can easily see under the light microscope. So that was the first virus that was, in fact, violating the classical criteria by VOF. You can see them. This is classical normal skew optics, and you can see, and you can even, in a way, guess some kind of a geometrical shape for some of those guys. Okay. So it's easy to see. The shock was not only the size, because of course, science was not only convinced by the size or the morphology of the thing. We had to do a little bit of sequencing to convince them that was a virus. And the first surprise, it was that the genome size was more than 1 megabase of nucleotides, 1 megabase of base pair. And at the time, the largest known virus was an algae virus that was only 330 kilobase pair, which is already quite a big virus, of course. So that was quite a big shock also. But the shock was, in fact, that was also the first virus that had, this is right here, that had many more genes and a much larger genome than many bacteria. And in that list, there are a lot of intracellular bacteria, parasitic ones, but some of them are not. we were able to, for example, cultivate Trophioma. We play in a rich medium also, Mycoplasma not intracellular. And many of those bacteria give you real disease, okay? And so that was a surprise that you can have a virus that has, in fact, more genes than many regular bacteria, I would say, parasitic ones for most of them, and of course, many more than some of those totally crazy little bacteria that we keep discovering with even more reduced genomes than the regular parasitic bacteria. But the very last surprise in that genome was that we found many, many components of the translation system, which was a real no-no for all the criteria. Okay, there is one thing that viruses don't do. They don't do translation. They leave that to the cell. Okay, and so we are very surprised to find that this virus encoded 4 amino acid tyrosine synthetase, and all the translation factors needed to end, to start, and to elongate the proteins. Okay, and this is very special because this type of enzymes are the most central component of the translation system, because when you think of it, this is the only component that knows about the genetic code, because this is the enzyme that makes the right connection between the right tRNA and the right amino acid. If you don't have that, you don't have any kind of genetic code. Okay, so that was quite a bit to amount for horizontal gene transfer, for example, because 4 of them was quite a lot, and so that was a real surprise. And immediately, the first hypothesis we put up at that time, if you believe that translation is the specificity of the cellular world, well, one possible explanation for those kind of viruses would be that they, in fact, have an ancestor that had translation. And if that ancestor had translation, it was some kind of a cell, an ancestral cell type, okay? And what we see, and what we are seeing today is the result of a genomic reduction, and what we see in that genome today are just the remnant of a previous more complete translation system. Okay? So that was the first hypothesis trying to explain the reason why we could find such big viruses with this type of genes in its genome. So a little bit of biology here, how those things works. Well, this is a Mimivirus right here, just contacting the membrane. And this is detected as possible food by the amoeba, because amoebas normally feed on bacteria. And we know now that the envelope of that virus, okay, is very much made of some kind that looks like murine a little bit, and probably also it tastes a little bit, it's sugary, it tastes like bacteria capsules, for example, for like spores. Okay, so immediately and also it has the right size. It has been known for quite a long time, people using latex bead in the 50s, that in order to trigger phagocytosis for a single particle, you need to have particles that are about 0.5 micron in diameter, and this is 0.7 micron in diameter, basically. Okay, so immediately you have phagocytosis by the amoeba, and the thing the particle gets into the vacuole, and as you can see, there is that very nice contraption here in the particle, which is only on one side, on one single vertex, so the particle is not, in fact, symmetrical. It's not a perfect icosahedron. And that thing's open up, and when it is in the vacuole, and because there is an internal membrane inside that particle, okay, this membrane can unfold and in fact fuse with the membrane of the vacuole, okay, and what was inside the particle gets inside the cytoplasm that way, okay. It's just by membrane fusion after opening of the particle, and you can really see here a nice synapse of the membrane here, okay. And so this was the first example of a virus getting in by phagocytosis and then vacuole fusion. So in fact, this is food poisoning for the amoeba. The amoeba believes that she's actually feeding on bacteria. And also there is something that is quite different from other viruses. There is no what we call normally eclipse fate. Normally you see something, the guy just disappear, and after a while reappear as new particles. In that case, in fact, this is a particle that is not yet unfolded. And this is the same at the inside of that particle. And you can see that the inside of that, the core of that particle is right here. And this core of the particle is going to get raised to what we call a virion factory. You can still see exactly the same dimension here. And what you see here are plaques of intense DNA replication and DNA transcription under the element. So this is, you can really see the virion factory here, which in fact exclude all the organelles of the cell, except it is recruiting mitochondria right around here, like here and here. Okay. And after a few hours, okay, your virus has become this. Okay, and you can see new particles emerging from this virion factory here. This is the amoeba, totally filled up with those new particles. This can go up to about 6 micron in diameter at the very end, occupying almost the whole volume of the cell. And here is the nucleus that is totally untouched by the infection. Okay, the virus doesn't care at all about the nucleus. Everything that is done in the cytoplasm of the cell. So this is the actual virus. In fact, this I can really see the virus as some kind of a transitory microorganism. In fact, that just gets ingested as a particle, developed totally into some kind of a new microorganism that is totally independent from the rest of the cell, except for the need for translation and ribosomes and energy and nucleotides, and makes more of viruses. Okay, more virion. We actually made that mistake ourselves because in the very first paper that we published on this virus, we mislabeled this as the nucleus of the cell, in fact. So that was never corrected, except for today. Okay, so that was the first paper. The second paper was the complete genome and all the interpretation we tried to make out of it, including the possibility that these things could come out from a fourth domain of life. You probably have heard of that. But also what was nice in having the whole sequence, at that time I was coming back from NCBI, I still knew Craig Venter pretty well, and he was starting to mount his fantastic database of metagenomics, the Sargasso Sea database, okay. And I got to see a quick look with that sequence, because what was very amazing also in that genome sequence is that a very small percentage, about 80% of the genes we found in that genome didn't look like anything. And we couldn't relate anything almost to the gene of the virus, to the regular database. And the only good matches we could find were in the Sargasso Sea database. So we knew there were relatives of this virus, Mimivirus, in the sea. So immediately we changed gear in the lab, and we decided to look for new virus of the same type in the sea to have a better understanding of exactly what we had in hand. Roughly about the same time, marine oceanographers and marine microbiologists were starting to understand. They knew that there were viruses in the sea starting in the late 50s. No, I think 70s, basically. But they were mostly phages, okay. But at that time, Willie Wilson and a lot of people working on some planktonic species like Emiliana Huxley, which is a very ubiquitous species of a small microalgae. You will see the picture later on. Those guys have blooms, absolutely immense bloom. Nobody knew exactly how those bloom appears. That basically form megatons of those things in less than a few days. But there was another mystery, is that those things also disappear very quickly. Okay, and nobody knew exactly why they would disappear. And Willie Wilson and Schroeder found that, in fact, this was just because they will catch cold. Okay, they will just get the flu, and they will die of virus infection. And it turns out that this virus, which is right here, so this is the Emiliana Huxley, a beautiful little protist here, unicellular protist with those limestone little contraptions, which are very nice, which are basically responsible for the cliff in Dover or Calais. Those are mostly 100% of those things, basically, about 400 million years. And those are killed by this virus. And this virus was already quite a big virus because they did the sequencing of that virus in 2005, and it would have been the largest known virus at the time if we hadn't skipped them with Mimivirus, 406 kilobases. So they knew that there were large viruses in the sea, and they started to understand that maybe those viruses might be very important for the regulation of plankton populations. Since then, we've been working on those viruses also. And there are other blooms, like the one that Phthisitis globosa, another algae in aptophyte, is actually doing, are also killed and regulated by this virus that is, in fact, also an even bigger virus right here in terms of genome. And we just recently sent out to GenBank another one, which is a little bigger than that is infecting. The name is Chrysochromulina ericina. It's another aptophyte, and this one is near 500 kilobase. So we know that there are a lot of large viruses in the sea, and they infect unicellular protists, but not human. And so we decided to develop that activity in the lab using the Assemble European program that gives you access as a non-marine biology lab, give you access to many marine stations throughout the world. And so we used the one in Chile because of the upwelling along the coast of Pacific of Chile is extremely biodiverse, one of the most biodiverse places in the world. So we decided to start going there. So we went there, the fun part. This is Randy. We are so amateur that we wanted to take a little bit of sediment, but we forgot to bring something. And so this is a bottle of water that we cut, so Randy could actually pick up a little bit of sediment from the bottom of about 20 meters deep here. And that was fortunate because from this little sediment pot came out Megavirus. Megavirus was an even bigger virus, a little larger in terms of capsid side, short hair compared to right here, shorter compared to Mimivirus, more military look. 1.8, almost 1.3 megabase, so a little larger virus, but definitely the same type of virus. And when we did the genomics of it, we were very pleased because, remember our hypothesis that we're in fact looking at genomic reductions, and in fact we had some translation system before that we are just losing, and we're very pleased to see that in larger genomes we found more of those amino acid synthetases. We found 7 of them. We found the 4 that we already found in Mimivirus and 3 additional ones, which was very much even more difficult to interpret just as horizontal gene transfer, because again, having 4 or 7 of those enzymes is not good enough. You need either 20 or nothing, okay? And since then, many other people have found relatives of those viruses, and in all cases, you find different numbers of those amino acid synthetases, mostly all the time, the same, plus 1 or 2, which are from the same subset, if you want. And the smaller ones only have 1. So this definitely seems, again, to indicate a process of genomic reductions, and seems to indicate that the ancestor of those viruses were some kind of a cell that has a translation system. So since the very beginning of this era, well, this was when we started, okay, this is Emiliana Huxley, and all those guys are members of the same family that we call now the Megaviridae or the Mimiviridae, okay, and seems to peak around 1.2, 1.3 megabase of DNA, and about a little more than 1,000, 1,100 genes for the largest of them, protein coding genes, right? This is definitely emerging a new family. Those are eukaryotes. This is a DNA polyp phylogeny. Those are the biggest, families of double-stranded DNA viruses, large ones in general. And this is definitely a new family, a very high bootstrap value, and that is in fact start to split into 2 groups, I would say. The very large ones, and all of those very large ones are the ones that are infecting Acanthamoeba at the moment, okay? And a group of smaller ones, which in fact are infecting other unicellular protists, mostly microalgae, and some other protists which are not photosynthetic, like Cafeteria or Borbergensis. So all those things are unicellular organisms, don't worry. And we even start finding them in totally mislabeled entries in GenBank. In fact, this was mislabeled as a one of an Eukaryote. In fact, this is a new member of that family that we now call Washington virus because it was found in Washington Lake in Washington State in the US. Okay. They have a couple of characteristic features. They all have a very strange repair enzyme, a mismatch repair enzyme called MUT-S, but they have the version 7, okay, which is only found in the mitochondria of octocorals. Don't ask me why. Okay. Those octocorals are the only organisms that have actually a MUT-S in their mitochondria, and those viruses, all of them have this gene. Okay. They also have an asparagine synthase, which is basically making asparagine out of aspartate, and so that's, I don't know why, but they all have this, and it's a very clear physiologic signal. And they also have that very thing that I will not talk about too much today, not at all, in fact. They have a virophage. A virophage is a virus of the virus. That is, many of those viruses, Mimivirus, Phaeocystis globosa, CEV, and organic lake virus, which was found in Antarctica. Those guys have a little bitty virus that is about 25 kb of double-stranded DNA virus that, in fact, is a parasite of their virion factory. Not of the amoeba, of the host, but of the virion factory. So basically, the guy penetrates with the big virus, waits for the big virus to put together the virion factory, and then use the machinery of the virion factory of the big virus to make its things, okay? And go out as a virophage. Okay? So this is very, very unique also in virology. So we're very happy. We continue to explore different places to find more of those virus guys to try to understand more their origin and their evolution. And so we went, basically at the same time, I went to Chile again to try to re-isolate actually new viruses of the same family, because at the time we thought there were only one family. And at the same time, I was invited to a meeting in Melbourne, and in La Trobe University, there was a pond in the middle of the campus, like here. And at the end of my talk, I had a plastic bottle, not something like that, okay? I went to the pond and I pick up a little bit of sediment from that pond and I bring it back to the lab. And we put those sediments in contact with amoebas, like we do all the time, and we started to see things happening, some new things replicating, being amplifying. And at first time, we started we call that new life form 1 and new life form 2 because we don't know what to expect. This is Mimivirus under the light microscope. This is a light microscope of the microscopy of the two other guys, okay? At once, we thought that we had a contamination because they were looking very similar, but a quick protein gel showed that they were different, so they were different isolate of whatever that was. And this is what they look like under the electron microscope, okay? Empty. This is basically nothing, except there is a very electron-dense sphere, a very complex, there is an internal membrane, lipidic membrane to those viruses, like for the Mimivirus, and a very complex three-layer tegument that very much look like what you can see in fact, amoebas when they make not spore, but they make cyst. When they are in a stress conditions. Okay. So and there is that nice osteal thing. So of course we extracted DNA of that thing, and the surprise was now we had a virus with a genome. It's a virus. You will see why. That has 2.8 megabase of DNA. Of it was about 2.5 was unique sequence and encoding about 2,556 coding for proteins. Okay. So this is just the two strands. This is the homology with something in the database, and this is the protein that are found in the proteome of the particle. Okay. So the particle is made of about 300 different proteins encoded by the virus. Okay. So what is that thing? Well, we didn't find any ribosomal protein encoded in that genome, and we don't see anything. We don't find any traces of dividing apparatus in the genome. We don't find any trace of ATP production. We don't see anything related to translation, so that must be a virus. But for the first time, we don't find any capsid protein encoded in that virus. Okay? And so, again, it was a very big shock. And the second shock was that it was even worse than Mimivirus. This is what doesn't look like anything else in the whole galaxy. Okay. 92%, I think, or yes, 94% of the genes don't look like anything. Okay. And it is possible that what we can actually see with some homology might in fact be due to lateral gene transfer from other things into that virus. So the true essence of this virus will be probably this and not this, which is probably horizontal gene transfer. Okay, so at this point we say, well, one explanation for this is that we made a bioinformatic mistakes, because in order to find those homologies, you need to translate according to the genetic code. Of course, if the genetic code that is used by this organism is not the natural one, the sequence you derive from the sequence are not right, and so they are not going to be blastable, they are not going to match against the database, okay? But fortunately, we had at least 200 or 300 of those proteins that we could see in the proteome of the particle that were encoded by the virus, and so we could predict those proteins, and we could see them in a tube and make proteomics out of it, okay? And yes, those guys do exist, okay? So we had the confirmation that yes, those proteins are translated according to the regular code, and so if they don't look like anything else, it's because they don't look like anything else, not that we made a translation mistake, okay? And so, again, in the protein that we know are in the tube, 83% of them don't have any match in the database. So this is not just a subset of strange proteins, okay? When you look at the other guys, so one is called Salinus, the one from Chile, because it was from the sea, and this one is called Dulcis, because it was from Australia, because it was in fresh water. Basically, looking like this guy looks like a subset of the big guys, but in fact, about 50% of the genes in this genome don't look like anything in these guys also. So already we had an indication that this family was probably quite broad, and that was probably not the only one that exists on Earth, okay? And so, but this one is only 1.9 megabase in terms of. So now, the new situation is now, when we thought we had reached an asymptote here with the Megavirus, now we are here. Okay. And so if you ask me what is the limit for a virus, I can't really tell you anymore. I don't have any estimate about this. This time, the virus is not only bigger in terms of genetic size than a bacteria, it's also bigger than some parasitic eukaryotic cells, like Macrosporidia. Okay. So now we have a virus. We have the virus world basically encompass both the bacterial world and the eukaryotic world in a way in terms of genomic complexity. Okay. So this is quite an amazing story. So what, a little bit of biology again. Well, everything starts the same, phagocytosis. The amoeba is so happy, thinks he has bacteria. No, the plug opens, okay? There is a fusion of the internal membrane of the virus with the membrane of the macule. What was inside that we don't see get outside and get into the cytoplasm. You can really see here, very nicely here, okay? Downloading or whatever is in there. But a big difference with Mimivirus this time, this is a regular nucleus of an amoeba, of a healthy one, and this is what happens to the nucleus of the amoeba, let's say 1 hour after the infection by the virus, okay? So definitely this virus do something to the nucleus. It's not a cytoplasmic virus, and we did that even better because after a while, the nucleus is totally dissolved, okay, and replaced by some kind of a I would say, invisible or not very visible virion factory from which we can see emerging new particles of that of Pandoravirus, Salinus in that case. One other crazy thing is that, of course, those things don't divide. And they form in a very strange way. They form like, it's like, making a bottle. At the same time, you fill it, okay? Because, you can see you start from what we call the ostial apex right here. And you start, doing something, and you finish here. But, it's not like most viruses in which you make the box first, and you fill up the box, okay? In that case, you make the box as you fill up whatever is inside, okay? So the size is 0.2, so this is about 1 micron in length, and about 0.6 micron in diameter here, okay? And at the end, this is what you get, okay? This is an amoeba full of those new particles that are going to emerge after the amoeba is going to explode when it's going to be filled up with those particles. So we know that the nucleus is involved for other reasons. First, we see that at least 10% of the genes in that virus have introns. So for this, you need the spliceosome, and the spliceosome, you need to be inside the nucleus. We also know that those viruses don't embark their own transcription apparatus. So they don't have any RNA polymerase, so they need to use the one that is in the nucleus. They encode one, but they don't bring it in the particle. Mimivirus does it. Mimivirus has everything on board, okay, as proteins. These introns are quite difficult to pick up because most of them remain in phase with the flanking exons. So try to pick up those when the protein doesn't look like anything, as you know. So we know that now there is many more of those because we have the whole transcriptome of this virus. But we know again that the protein doesn't incorporate any transcription machinery, so it has to go first into the nucleus, and nobody really knows how in less than about 15 minutes you can bring about 2.8 megabase of DNA through a contamoeba into the nucleus so quickly. This is very, for the biophysicists, this is a real mystery too. So what are the Pandoraviruses compared to the Mimivirus? Well, nothing. They are totally independent of the rest of the other big families of DNA viruses we've seen. So the Megaviridae are here, with the small one and the big ones. And Pandoravirus, the two guys, are all by themselves, okay? We now have 5 of them and one from an independent lab, and they are all quite different. So we call them Pandoravirus because we thought that definitely opened a Pandora box in terms of what we thought a virus could be, and also they look like a Pithos. And in fact, the Pandora box of the legend was not a box at the beginning. It was in fact an amphora. Okay, it was a mistake in the translation that makes the Pandora a Pandora box. Okay, so this is and this was a nice cover in science, but. Now we're trying with our hypothesis because, okay, we Mimivirus was a typical, very cellular ancestor, but this guy is totally different. He's even bigger. So if we want to use the same genomic reduction IG, we have to say, well, there was a different ancestor from those two guys. Okay, so now we are not in a 4th domain of life, but we are now in a 5th domain of life eventually. And so the idea we put up is that probably before Luca, much before Luca, this is a totally wild speculation, but that was actually confirmed very recently, about 15 days ago, by a work by Gustavo, what's his name? Caetano Anoles from Illinois University. We believe that, in fact, those viruses, those very large viruses, DNA viruses, are coming from a time where there were many, many different type of ancestral protocell lineages. Okay, they were all competing in evolution at the time, a long time before Luca. Okay, one of that lineage gave Luca and gave what we have today. Okay. And those guys, we call them the losers, lose the race, okay? And they only had one possibility to survive a little bit, was to become parasite of the successful one. Okay, so we believe that many of those viruses are in fact coming from very, very ancestral lineages. Okay, that have survived partially as parasite of those guys at the time when, for example, the eukaryotes have been emerging after that influx of, of the mitochondria into the cells, probably an archaebacteria. By the way, you know that now we are not a real domain anymore. We are just part of the archaea. Okay, this is the new domain. We are just archaea. So you think this is crazy, but in fact, as you may or may not know, a very robust work has been done by a couple of people in France that showed that probably there were some kind of a pre-metazoan that were existing 2 billion years ago, when we thought at the beginning that some metazoans were invented here. Okay, so what we know about what really happened a long time ago is still very much I would say science fiction. So the next story is, after I saw that paper by David Gilishinsky, who unfortunately died right after this paper was accepted, his group had been doing microbiology of permafrost for a very long time. And I look at that paper and say, well, if those guys are capable of rescuing a plant from permafrost, why not try to rescue viruses? Okay, so I just called them, asked them if I could get a little bit of a permafrost sample, the same that they got the plant from. And they say yes, and we did as we do usually. We took that sample, and we put that in contact with amoebas. So first, where is it coming from? Not a place when you go very often in vacation, okay, as you can see right here. This is a very strange place because, in fact, permafrost there, which is the permanently frozen ground, basically peat, if you want, is carved by a river here. And in fact, instead of having to go from the surface to pick up a certain date, you can actually go sideways. And so you eliminate all the possible accusations of contaminations or whatever, because of course when you go from the surface you will contaminate your sample from whatever is on the surface, and so the younger layers. Actually, in those places you can go up to 1 million years old permafrost. Okay, and so we went this and again put that sample in contact with amoebas, and we got that. Okay, this is again regular microscope pictures. And so at first we had a new Pandoravirus from the time. But when we looked at it under the electron microscope, it doesn't look at all the same, about the same shape. It was even bigger. This is now 1.5, 1.2 to 1.5 micron in length, 1.6 micron in diameter. That very strange plug, when you cut the plug like this, you get that very nice honeycomb type of figures. It was, again, totally a different virus, again with a lipidic membrane inside. But this time, during the infection, the nucleus stayed totally intact. This is a nice, happy nucleus of the amoeba here. So this guy is, again, a purely cytoplasmic virus, okay, like the pox viruses or the Mimivirus. We did the sequencing of it. The same story, a little more known genes here. A lot of 2/3 of totally unknown genes, okay, and a little bit of genes probably picked up or with some very ancestral relationship with bacteria, virus, and eukaryotes. Again, it's a very different type of virus from Pandoravirus. Pandoravirus is GC rich, this one is AT rich, like the Mimivirus. A very small genome. In fact, for us, it's only 610 kilobase of DNA for such a large vehicle, which is very strange. So there is no correlation between the size of the particle and the size of the genome. A couple of nice pictures to show exactly the complexity of the plug here. We call that the cork, okay? And at one point in the development of the virus, we have those very nice rectangular shape before it gets rounded and the teguments came in. And the teguments had a totally different structure than the other one. The tegument of Pandoravirus is more like layers like this, and this one has layers like this. Okay, so it's totally different virus, which is actually confirmed by the genomics. Because when we do the phylogeny of those viruses now, so now here we have the Megaviridae, where is the Pandora virus are still here by themselves, I think. Yes. And strangely, Pithovirus, but that could be an effect of horizontal gene transfer, has some affinity with a group of viruses that are known as iridoviruses and the Marseille virus, which are also viruses that can infect Acantha amoeba. Okay, so this was a big buzz because we were, in fact, waking up virus from the past. In fact, 30,000 years ago, this was the extinction of Neanderthal, which means that, in fact, there is some possibility of a continuity of infection disease between Neanderthal and us. To us, that was showing that, in fact, you could have totally two different viruses that would have basically the same shape. So, where we are thinking of viruses always as a thing which are mostly with geometrical shapes, whatever, we thought that this could be a way to think that this is a different type of vehicle in which we may put as much variety of viruses than you can put in other regular geometrical boxes, for example. So we were maybe discovering a brand new branch, I would say a brand new internal domain of virology here. Okay, so the last one is this one from the same sample. We knew from the beginning that we had something else, and so finally we succeeded in cloning it. And this is published a month ago. We call it Mollivirus. A big buzz again in French TV, in CNN, in a popular journal in France, cartoons in Charlie Hebdo also, which are very proudest. Again, no problem to see it under the light microscope, okay? A round face this time, again, internal lipidic membrane, a little opening here, which is the one from which everything that gets from inside outside in the amoeba. Again, a totally different virus. That is, it's a different type of organization for the viral genome. It's only 650,000 base pair, only 500 protein genes. It's nucleocytoplasmic. It has probably two phases, and it replicates in 6 hours in Acantha amoeba and doesn't explode the amoeba, but probably gets outside using exocytosis, which is very strange. So now we have 4 families, totally different families of giant viruses, 2 of them from the past. We know that there are modern pithoviruses. We've seen people that have seen them in Acantha amoeba about 10 years ago, but thought they were bacteria, endosymbiont, so they didn't thought it was viruses. There is also the Marseille virus, which are quite big viruses, but not that big, that also infect amoebas, okay? And so we are busily, of course, characterizing all those viruses, doing everything we can to get more information about about their physiology. Those are the Mimivirus, Pithovirus, Pandora, 200. So those are basically at the same size, the same scale for the 4 viruses. This is the same thing under the scanning electron microscope. So you see the Mimivirus here. You really see the opening on this guy, and I like the amphora shape of this one very nicely. Okay, all of them have the same infection strategy. Get into the bacteria, gets into the amoeba by pretending being a bacteria, trigger phagocytosis, open up lipidic membrane fusion, download the virion interior, and then do different things depending on if they are, in fact, I would say transcription competent or not transcription competent. Okay, so 2 of them are, 2 other guys don't. The thing that is important is that the one that we rescued from the Permafrost, okay, one is pox-like, that is cytoplasmic, and the other one is like herpes-like, if you want, nuclear. And so that means that the fact that you are cytoplasmic or nuclear doesn't correlate with the fact that you can actually survive 30,000 years in the permafrost, which, of course, has some implications about infectious disease from the past. Okay, all of them have all that huge compartment of genes that don't look like anything, which in fact is creating a lot of fuss in terms of surgical biology. Where are they coming from? So as I told you, there is a lot of different, totally opposite hypotheses. Like Kunin will think that all the genes, all the viruses started very small and are getting bigger, and we believe the total opposite. And with Cayetano I know less also that we believe that they started very big and they are just reducing like all parasite. We don't see any parasite. Genomic reduction is very classical among parasite. We don't see why viruses would be that different. It's a new discipline. We call that environmental virology. Up to now, viruses were always found in the context of a disease because that was so easy. With the disease, you get both the host and the virus. Okay. When you get the virus without the host, it's always a difficulty. So that's why metagenomics doesn't really help you to find new viruses because you get sequences, but if they don't look like anything else, how do you know you have a new virus? So you really need to isolate those guys. At the moment, we are just using Amoeba as a system because it's still quite efficient to find new viruses. If we don't find any more, after a while, we'll shift and look for other hosts. Just to thank all the people that have been involved in that work for the last 5 or 10 years for some of them. Most of that is the lab. The lab is half bioinformatics, genomics, and wet lab for the characterization of the viruses. The proteomics is always done in Grenoble, which is one of the best places in Europe to do proteomics. The Permafrost samples are given to us by the people in Moscow. And this is the Parc des Calanques. And I want to salute Chantal Aberger, who is the vice director of the lab, who is doing the whole wet lab work. And I like very big viruses because you can put a lot of text inside the Thank you.
Jean-Michel, I can say my mind is very definitely expanded. Questions?
Thank you very much. Fascinating. So a virus is small and is very effective in doing the cycle. I'm wondering, what is the evolutionary purpose for these guys to be so big?
No, you've said everything. They are a total anomaly. If you can go by with 3, 4, 5 genes, a little box, and be extremely efficient like the HIV or even papillomavirus or whatever, why would you become a huge thing? So
there's got to be a purpose.
So no, I think there is no purpose. I think the only reason, the only way you can truly explain this by what we know in terms of evolution is by genome reduction. You cannot invent a small thing and make it big. But yes, you can think that those guys started very big, and for some reason, let's say in amoebas, they evolved very slowly. So all the other guys have shrunk extremely rapidly and have become what they are today. Like we can even see that in the family Mimiviridae, when we already see that there are 2 different branches, one that seems to evolve very slowly, the guy that infects Amoeba, and the guy that seems to evolve a little more quickly, which, by the way, are viruses that infect blooming algae, which are the one that, And so in that case, you have fantastic production of viruses, and so you have a lot of places for evolution and changing and eventually reductions. In the case of amoebas, we don't know how often those guys divide. Amoebas are everywhere, but maybe the virus divide once in natural conditions once a century, whatever. I don't know. We don't really know. Also, those viruses have all the possible DNA repair mechanism that is known to man. Okay, so they are extremely cautious with their genomes. Okay, so I think the only possible explanation, if we don't want to change everything we know about evolution, is that they started big, and they are reducing. Some are reducing quicker than others. Okay, and we can see that. We have those viruses which have everything but ribosomes, but they still have a little bit of amino acid transferase. Okay, then you have all the viruses that have kept transcription, and they use it, and they are cytoplasmic viruses. Then you lose transcription, you keep DNA polymerase, you keep replication, then you lose it, and then you end up as being, papillomavirus or adenovirus or those type of things, for example. But there is some kind of a gradation. Those guys are absolute parasites, but what we are discovering is that, in fact, there is a gradation in absolute parasitism, which is an oxymoron in a way, but apparently, yes. You just, basically, because it's totally crazy when you think about it. Why would a virus keep transcription when it has to go to the nucleus anyway to transcribe in the first place, because it has on board its transcription proteins? Okay. So everything's there. So what's the need? And in fact, we have evidence for Pithovirus, for Pandoravirus. In fact, the DNA polymerase is made much before the transcription of virus is in place. That is, in fact, the DNA polymerase of the virus is transcribed, which normally is a late phase type of protein, is transcribed by the transcription apparatus of the cell, not of the virus. So what is the need for having those? That's very strange. No, I totally agree with you. I have no explanation for anything.
Yeah, this might be also silly. Is there any evidence in the actual antamoeba genome of horizontal transfer from these kind of viruses? In other words, do we find genes which don't fit anywhere actually are in the antamoeba?
No. We've done. We've done. Of course, we've done the genome. We are using. We are using Acantamoeba castellani, and we've redone the genome ourselves. We've done the transcriptome, so we know that perfectly well. No, there is absolutely no gene transfer between the two of them. If there are evidence, sometimes there are evidence of the virus transferring some genes to the host. The converse is much rare. It's much rarer than that, in fact.
So I'm wondering how common they are. So you've documented, you tell the story of all the places you've looked and found them, including in university ponds and so on. How many times have you looked and not found them?
Now that we have a protocol in place, I would say that half of the time, when we sample everywhere, besides the lab, we find at least Pandoravirus, Mimivirus, and Mollivirus. The Pithovirus, we haven't found them easily yet, and the Mollivirus, we just published the first one a month ago, so I cannot tell you. But no, this seems to be everywhere, and I have a map to show where they have been found. They are planet-wide. They are in Australia, in Madagascar, in the US, in Canada. They are everywhere.
Yeah, I guess you just said that you use the Acanthamoeba in the lab as the target host for these isolates from all around the world. Could you speculate from that that probably isn't their native host, and so these things maybe have rather Catholic or broad host range as parasites?
We try to, in fact, Acanthamoeba is a fantastic host for isolating viruses. It's a terrible host to work with them because there is nothing works with them. Okay, there is no genetics in place. There is no transformation ways. There is no way of doing anything. Okay, so we try to switch those viruses to a different host, like Dictyostelium, right, for which we have everything. Okay, no way. We try to isolate virus directly on Dictyostelium from the same places. No way. So yes, we start to believe that Acanthamoeba and other Acanthamoeba and other amoebas are very special in that respect. And as you know from the bacterial world, we know that Acanthamoeba is the home of a zillion of endosymbiotic bacteria. Any bacteria that you can think of can eventually survive for quite a long time in Acanthamoeba. So for some reason, there are a very good niche for those viruses. And we don't really care about this. If there are a fantastic way to explore the unknown virusphere, that's fine with us, okay. But yes, a second set of the research would try to understand why this seems to be so special about viruses and bacteria, actually. Because Acanthamoeba doesn't work the same way either.
One more question down the bottom.
I wondered if the size is about transmission. So if they have to be a mimic of a bacteria to be the right size,
yes, absolutely,
in order to get in,
yes.
Does that then, as long as you're already that size, you may as well
yes.
maintain a lot of DNA?
But the size of the particle is totally, except for Mimivirus, and even for the case of Mimivirus, the DNA in Mimivirus is probably 10 times less compacted than in classical phages, for example. So there is plenty of room. You could pack about 5 megabases of DNA in Mimivirus. In Pandoravirus, you could pack, or in Pithovirus, you could pack 35 megabases. So you can pack a yeast genome or whatever. So there is no correlation between the genome, which is probably coming from the genome reduction type of evolution, and the size of the particle that needs, as you say, to stay big enough, because in those dilutes, especially in the sea, when things are extremely dilute, except when you have blooms, okay, it's not very often that a virus is going to encounter its host, okay? And so you really have to be certain that the unique time is going to happen, the infection is going to be productive. So it's very important, you're right, it's a very strong incentive for the virus to keep a very visible particle, absolutely. And that may explain a little bit of that. Absolutely.
I think we'll stop there.
Thank you.
Very much again.